Hello and good evening to Dr. Chung. My name is Darshini and today I'll be presenting my proposal poster with the title Immunomodulatory Effect of Bifidobacterium breve on Experimental Allergic Rhinitis in BALB mice. Allergic rhinitis, or AR, has emerged as a major public health problem worldwide. It is a complex, multifactorial disorder involving both innate and adaptive immune responses. It is also characterized by an elevation in the serum immunoglobulin E, or IgE levels. Probiotic bacteria has an ameliorating effect on human diseases, including preventing allergic disorders, aiding in Th1 and Th2 polarization, as well as inducing appropriate regulatory mechanisms. B. breve has been shown to be the most effective in reducing allergic responses such as allergic rhinitis via Th1 and Th2 cytokine level analysis. However, the optimal dose for this disorder has yet to be determined. There are two objectives for this study. That is to investigate whether microbial induction of regulatory T cells, or TREGs, and adjustment of Th1 and Th2 responses by Bifidobacterium breve are associated with protection against allergic inflammation, as well as to identify the optimal dose response of Bifidobacterium breve administration. For the methodology, we have a few steps here. Firstly, 60 BALB mice were housed in suitable conditions with sufficient living needs as standard and distributed into 6 treatment groups. The mice were then sensitized with intraperitoneal injections with PBS or OVA, followed by daily nasal challenge via nasal drops. The B. breve strain was administered to these mice via oral gavage daily from day 14 until they were sacrificed on day 56. The observation of nasal symptoms was recorded by counting the number of sneezes by these mice for 10 minutes every day. ELISA, or enzyme-linked immunoabsorbent assay, was performed on the samples. The serum samples were collected, clotted, and centrifuged prior to the analysis. The total immunoglobulin levels were then calculated. Cytokine levels were also determined by Luminex kits used in this research, and other tests such as histopathological, splenic tract, and statistic analysis were carried out at the end of the research. As for the results here, the results suggest that B. breve intervention reduces the production of OVA-specific IgE, as well as being able to relieve type 1 allergy disease in mice. The administration of B. breve at doses of 10 to the power of 10, 10 to the power of 9, and 10 to the power of 7 CFU significantly decreases the OVA-specific IgE levels, as well as sneezing frequency, and nasal mucosal epithelium was protected from the local allergic reactions, while sneezing frequencies was unchanged at 10 to the power of 5 CFU. However, a lower dose of B. breve could only slightly relieve the allergic reactions, as lower dose equates for lower OVA-specific IgE levels in the body. Oral administration of B. breve has an anti-allergic effect by inhibiting the Th2 response and inducing the activity of CD4 and CD25 and TREGs without evoking the Th1 response. Administration of B. breve at a higher dose significantly reduced the sneezing frequency combined with decreased serum IL-4 and OVA-specific IgE levels, and increased the CD4, CD25, and TREGs in the spleen, and nasal mucosal epithelium was protected by from local allergic reactions as well. That is all for our research. Thank you very much.